So what I then wanted to talk to you about is the Neanderthal genome, and particularly how the Neanderthal genome at least partially live on today in many of us who are here tonight. So what I also then wanted to talk to you about is how we can get a perspective on the genomes of people who live today by comparing it to the Neanderthal genome. So I wanted to then start out by reminding you about what you all know, that our genome, our genetic material, is stored in almost all cells in our body, in the chromosomes, in the form of this famous DNA molecule, the double-stranded DNA molecule, and it's stored twice, essentially, once on each strand in the sequence of these four bases, A, T's, G's, and C's, and G's. And it's stored twice, and that's important because when a cell divides, these two strands come apart, and new strands are synthesized with the old ones as templates. And this process is very, very exact, but sometimes errors are made. Say, a wrong base is built in here, and if that's not repaired before a new cell division happens, it will become a mutation. So, although this is very exact, every new baby that is born has something like 100 or 150 new mutations that is not there, neither in the father nor in the mother. And you see these mutations, the effects of these mutations, in the form of differences in DNA sequences that you can observe among people in this room, for example. So if you compare two genomes, you will find a difference approximately every 1,000 base pairs. And these mutations rain down as a function of time on the genome. So if you take in a chimpanzee, if I can have the next slide, if you take in a chimpanzee, you will find a difference approximately every 100 base pairs. And if you're now interested in the history of these genomes, you can depict that history in the form of a tree like this, where these two human DNA sequences go back to a common ancestor quite recently, and much further back is there a common ancestor shared also with the chimpanzee. And as you know, the human genome is composed of a bit over 3 billion bases. So there is a lot of information there to reconstruct the genetic history of our species. So if we now do that and we focus on the variation among humans today, what one has then found out over the past 20, 25 years is that most genetic variation among humans is found inside Africa, among Africans, and less variation overall, although many more people live outside Africa than inside Africa, of course, there is less genetic variation outside Africa. And not only that, most of the DNA sequences you find out here have close relatives inside Africa. But there is then a component of the genetic variation in Africa that you don't find outside. And the interpretation of that is that modern humans, the direct ancestors of everyone who lives today, evolves in Africa, accumulates genetic variation there, and a part of that variation goes out and colonizes the rest of the world. And with some genetic tricks, you can also estimate approximately when that happened, and that is in the order of 100,000 years ago. So that's a genetic basis for the recent African origin model for modern humans. But there is, of course, a problem with this model, and that is that when modern humans then come out of Africa, starting about 100,000 years ago, there were other forms of humans around. Most famously then, there were the Neanderthals in Western Eurasia, and there were other forms of now extinct humans in Eastern Eurasia that are less well known. So we are then interested in these Neanderthals since over 20 years in our group. And the Neanderthals, here's a reconstructed Neanderthal skeleton on the left compared to a present-day human skeleton on the right. The Neanderthals are then these robust forms of humans that appear in the fossil record about 400,000 years ago and live in Western Eurasia until about 40,000 years ago when they become extinct in connection with that modern humans appear on the scene. And paleontologists, and you may then ask, why should we be so interested in Neanderthals? And there are several reasons for that. But I would point out particularly two here. And the first one is that Neanderthals are the closest evolutionary relatives of all present-day humans today. So if we should define ourselves genetically as a group, it's them that we should compare ourselves. What genetic changes do we have that they did not have? There are other reasons also for being interested in Neanderthals. One could, of course, be that they disappeared rather recently, 3,000 generations ago, and you may ask, what happened to them? So one has discussed that last question in paleontology in several decades, and there are different opinions about that. On one extreme, people who would think Neanderthals are the direct ancestors of Europeans today, so 100% continuity from Neanderthals to present day. To another extreme view saying modern humans come out of Africa, totally replace Neanderthals with 0% contribution. And you could, of course, imagine anything in between here, smaller or larger contributions. So the first chance we got to test this genetically came in the early 90s when we got access to the first Neanderthal, not just any Neanderthal, but the Neanderthal that was found in 1856 in Neanderthal and gave its name to this group of humans. So we got a sample from the upper arm here. At the time, we needed quite a lot of bone material to extract DNA. And we could profit from, by that time already, almost 20 years of experience on how you extract the DNA from old bones, how you work to avoid contamination by tiny amounts of DNA from yourself into your experiments, and so on. And we focused on a small part of the human genome, a tiny part, the mitochondrial genome, that is inherited from mother to offspring and exists in many copies per cell, hundreds of copies. So it's a much bigger chance that some fragments of the mitochondrial genome would survive than fragments of the nuclear genome, of which we have only 2 copies, right? One from our mothers and one from our fathers. So we reconstructed this little part of the mitochondrial genome and estimated such a tree as I showed before. And to our surprise, we found that the mitochondrial genome of the Neanderthal here went back far, over 500,000 years ago, to a common ancestor shared with the mitochondrial genomes of present-day humans. And all present-day humans go back to mitochondrial ancestor here between 100 and 200,000 years ago. So it's quite clear there are no people on the planet today that run around with a mitochondrial genome inherited from a Neanderthal. So in that sense, it is total replacement here. But it's also clear that the mitochondrial genome is a very tiny part of our genetic inheritance. In many, many copies here, but a tiny little part. The full picture of our genetic history is in our nuclear genome inherited from both mothers and fathers. And I think I'm on the published record in the order of 8-9 years ago saying we will never sequence the nuclear genome of a Neanderthal. There's too little of it there, it's too degraded, it's chemically modified. And I think that just goes to say that you should never make such negative predictions in science, because you're generally proven wrong. And it's generally technology that comes along and proves you wrong. And in this case, it was very clear. It was high-throughput DNA sequencing that came along in the early 2000s. The ability to sequence millions and billions of DNA sequences quite rapidly and inexpensively. So what you could start doing was to extract the DNA from a fossil and not look for any particular part of the genome. A bit louder. Maybe I should even take this in my hand, actually. Because this is... Let's see. Okay, let's do it like this and see if it works better. So what you could then do is extract all the DNA, simply sequence all the DNA in a fossil, make your little database here, and start comparing it to the human genome, to bacterial genomes, and so on. And the first place where this worked was in this site in Croatia, a beautiful site, Vindija Cave, and from this bone that's about 40,000 years old. And the first thing you notice when you then look at DNA fragments here is that they are very short. If you extract DNA from me today, you can easily get fragments of 20, 30,000 base pairs. Here the average size is 50, 60 base pairs. It's tiny little fragments. Another thing you will notice is that only a small proportion of all the DNA actually comes from the Neanderthal. The vast majority comes from bacteria and fungi that have colonized the bone when it was deposited in the cave. So we were very lucky then to get funded for 5 years to improve the methods with which we extract the DNA from the bone and convert it into a form that you can feed into the sequencing machines. So we got a lot better on this, and we looked at a lot of different sites and bones to find the best ones that contain the most Neanderthal DNA. And we focused in the end on 3 different bones from 3 different Neanderthal individuals from that site in Croatia. And then sequenced a little over 1 billion DNA fragments from them. Most of them then from bacteria and fungi, but what we then did was to match these fragments to the human genome, so to see where they fit, taking into account that they have typical errors. And in 2010 then what we had was a little over half of the Neanderthal genome, so we could begin to ask questions. And one of the first questions we were interested in was this question: what happened when modern humans came out of Africa and met Neanderthals? Did one mix with each other or not? So one prediction, if Neanderthals would have mixed with modern humans, would of course be that Europeans today would share more genetic variants with Neanderthals than people in Africa, since there have never been Neanderthals in Africa. So it's this idea here: if there is total replacement, the Neanderthal will be equally far from a person in Africa today as from people in Europe today. Whereas if there, if there is a contribution here, then on average a Neanderthal will be closer to Europeans than to Africans. So we had the Neanderthal genome, so we went ahead and sequenced 5 other present-day people, 1 person from Europe, 2 from Africa, 1 from China, 1 from Papua New Guinea, and did a very simple test. So we compared 2 present-day people that live today to test this idea, just 2 Africans, find all positions where they differ from each other. We then take the Neanderthal genome and see how often does a Neanderthal match one present-day human or the other. Since Neanderthals have never been in Africa, there is no reason to assume that the Neanderthal would be closer to one African than another African. And indeed, when we look at this, there is, statistically speaking, equally often matching one as the other. But this was then different when we took the European individual and an African individual. Then, to my surprise, I must say, we found that there was more matching, statistically speaking, to the European individual than the African individual. Even more surprising was the fact that when we took a Chinese individual, we again find more matching to the Chinese individual than to the African individual. And when we take the person from Papua New Guinea, a place where there for sure has never been Neanderthals, we find again more matching to the Papuan individual than the African individual. So there was a sort of surprise that we found a contribution from Neanderthals, not only in Europe where Neanderthals had lived, but also in China and in Papua New Guinea where Neanderthals had not existed. So the sort of explanation for that we suggested, and that has since been borne out by work from our group and from other groups, is to say that when modern humans come out of Africa, they presumably pass by the Middle East. So if those early modern humans mixed with Neanderthals and then went on to become the ancestors of everyone outside Africa, then they could carry with them, so to say, this Neanderthal contribution out to parts of the world also where Neanderthals had not existed. To the extent that people today that have their roots outside Africa then carry, sorry, go back a slide. No, go back a slide, please. Carry in the order of 1 to 2% of the genome from the Neanderthals. So when we published this in 2010, there was soon a lot of follow-up work on that from the other groups in science. That was very sort of satisfying. But what also became a surprise to me was how interested the general public was in what we did. So we immediately started getting a lot of mail from people who self-identify as Neanderthals and volunteer to send samples of blood and hair to us. And after a while, I started noticing a pattern in this correspondence. It was mainly men who wrote to me and suggested they were Neanderthals, and very few women who wrote and say they are Neanderthals. So when I sort of presented this to my group, they were very critical of my sort of experimental work there, and they sort of said, well, this is just because women are more interested in less interested in genetics than men, so the men will read the papers and write to you. But I went back to my emails and letters and found that that's not the case, because there were actually quite a few women who write and say they're married to Neanderthals, whereas there are very few men who claim they're married to Neanderthal women. So this, of course, is something that is very interesting to a geneticist and that we want to study more. However, we also do other things and count emails. And one is to study many other sites in the world, particularly in Siberia, a particular site called Denisova Cave, where Anatoly Derevyanko in Novosibirsk excavates since many, many years and find amazing things. It's at this beautiful site in southern Siberia. So in 2010, they found this little toe bone. When we analyzed this bone, it turned out to come from a Neanderthal. And by then, our techniques had developed. Now this one seems to have died again. Here we go. To the extent that we could then go from the genome we had then, where we just had covered a little over half of the genome with fragments, an example here, to a genome where we had covered every site many times over. So we actually now, since 2 years, have a Neanderthal genome where every position to which we can map these short little fragments are covered over 50 times. So we have a genome of a quality even better than what we would determine from a present-day person today. So what we can do is to, for example, look at different individuals on one chromosome here. These are different present-day people, and in red are the fragments they have inherited from Neanderthals. And you will see that people often carry different Neanderthal fragments, but on average per person it's then 1 or 2% of your DNA that come from Neanderthals if your roots are outside Africa. So you may then ask, is that a lot or is it a little? It's sort of a funny question, but one perspective on that perhaps can be to say, if you think about your own family tree, say this is you of course have 50% of your DNA from your mother and father, about 25% from your grandparents, then there's 12, there's 6, and it's 3. And if you go back 6 generations in your tree, you're down to about 1.5%, about as much as you have from the Neanderthal. So it is as if you had a Neanderthal ancestor among your 64 ancestors 6 generations back in your family tree. But it's differently distributed, of course. It's not in big pieces, it's in small pieces. So you can also ask how much of the Neanderthal genome still walks around today on 2 legs in different people today, because you can, of course, go from different people here and see how much can you puzzle together of the total Neanderthal genome from living people today. And that's a bit sort of unclear still. It depends on how many individuals you sequence, but somewhere in the order of at least 40%, probably about half the Neanderthal genome, is still there in people today. But this is not the only amazing thing they sort of found in the Denisovan cave. In 2008, they also found a bone, and I don't know if there are archaeologists here. I should perhaps be careful about what I say. But I think Western archaeologists often have a tendency to talk down on Russian archaeologists and say they excavate like we did 30, 40 years ago here. They just look for the big things, etc. But in 2008, they found this tiny little bone, and I think one must agree that I'm very skilled to realize this might come from a human individual. It's a part of the last part of a pinky of a child. And we managed to sequence the genome from this tiny little bone, and we're very surprised then to find that this genome was not a Neanderthal. It shared a common ancestor with Neanderthals, but far back in time. So we realized we had found some new group of humans, and after some deliberation, we decided to call them Denisovans after the Denisova cave, just as Neanderthals are called Neanderthals after Neander Valley, where they were found. And they are indeed rather distant from Neanderthals. So if you look at present-day humans here today, the deepest split among human populations today is in the order of 100,000 years. If you compare Neanderthals to these Denisovan guys, that's about 4 times deeper. So around 400,000 years then. One can ask, have they contributed to present-day people? And indeed they have, surprisingly, particularly out in the Pacific. So although we find the bones here in Siberia, there's the individuals today that carry most Denisovan DNA, up to 5%, is in Papua New Guinea, Aboriginal Australians, and so on. So if you should just summarize this sort of first part of the talk, first 2/3 of the talk maybe, about what we think about the origin of Neanderthals, Denisovans, modern humans from studying genomes. We think that Neanderthals and Denisovans have some ancestor in Africa more than 500,000 years ago. They come out of Africa. They evolve in Western Eurasia to what we call Neanderthals, and in Eastern Eurasia to what we call Denisovans. There were clearly other groups of humans there too. We don't know where the border between these really are, but we know that here in Siberia, at some time there were Neanderthals, at some time there were Denisovans there. Then modern humans appear in Africa, start spreading out of Africa and out of the Middle East, seriously only about 50,000 or 60,000 years ago. They mix with Neanderthals in the Middle East for the first time, continue to spread, mix again. For example, the ancestors of people in East Asia have slightly more Neanderthal DNA, so there's probably additional mixing there. There is mixture with these Denisovans then in Southeast Asia somewhere. One continues out into the Pacific, and then these other forms of humans disappear but live on then in the genomes of people today in the form of a Neanderthal contribution here and a further contribution in the Pacific here. So I wouldn't be surprised if there were also mixing going on at other places, for example in China, and for example in Africa, when modern humans spread across Africa. So a message here is that we have always mixed, at least a little bit, so to say, between different human groups. And we got a recent reminder about that is the case from this site in Romania. It's a cave site where cavers a few years ago found this mandible of a modern human. It is around 40,000 years old, so it's one of the earliest modern humans in Europe, and lived at a time when there were Neanderthals around. So we were, of course, very curious, had this individual mixed with Neanderthals or not? And it then turns out that if we compare, say, on one chromosome here, these are a couple of other modern humans on this chromosome, in color, the pieces that come from Neanderthals. And if we now look at this 40,000-year-old present-day modern human, we find a lot more Neanderthal contribution. And in fact, where it's black here, we have no information, so it seems clear that there is sort of a solid piece of the chromosome here that is purely of Neanderthal origin. And you can look across the whole genome, all the chromosomes like that, and you find 7 such areas with big, big pieces coming from Neanderthals. So you can then estimate in a family tree for this 40,000-year-old individual here, how far back was there a Neanderthal ancestor? And it turns out that it was either 6, 5, or 4 generations back in time. So it's quite amazing. We've sort of almost caught a modern human in the act, so to say. This individual had very shortly back in his family tree a Neanderthal ancestor. So we know there has gone mixing gone on there too. So we've clearly sort of disproven this total replacement. We have a bit of contribution from these groups to present-day humans, but the big picture is still that the variation of modern humans come out of Africa. So we sort of like to call this model of modern human origins a leaky replacement. The big picture is one of replacement, but there is a contribution from these earlier forms. Now, for the last 10 minutes here, what I wanted to do is shift gears a little bit and talk about what functional consequences may this Neanderthal contribution have to people today. And it's now beginning to come out more and more information about that. And I'm just going to give you a few examples. So with regard to Neanderthals, you can then sort of go over the whole genome here of people in Europe. You can look for regions where many people have it. Not just 2-3% of Europeans have something, but there are parts where 67% or 70% of people have it. So these are regions that may have been positively selected, may have been of an advantage and risen to high frequency in Europe and other regions, and sometimes same regions in Asia. So one example of such a region turns out to be a risk allele for type 2 diabetes, so the type of diabetes you get at old age. This risk allele is particularly common in Asia, up to 25% or so of people have it. And it turns out that that risk variant of that allele comes from Neanderthals into present-day humans and have then risen to high frequency. You may then ask, how can it be that something that gives you a disease comes from Neanderthals and rises to high frequency? It should be selected against. But I think it's sort of a good example of that genetic variants really interact with the environment to either give you a disease or be of an advantage. I think there is good reason to speculate that genetic variants that give you diabetes today, when we have ample nutrition through our whole lives, may be of an advantage in a situation of starvation. So it may be some Neanderthal adaptation to starvation that we see here that came in and were of an advantage in the past. You may ask, have Denisovans also contributed to present-day people in an important way, and there's big evidence that they indeed have. In Tibet, populations on the high plateau are adapted to living at high altitude, where there is little oxygen in the air. It was already known that it was particularly one variant of a gene called EPAS1 that's important for that adaptation. And a group in the US have found out that this variant, that is in 80% of people in Tibet, actually turns out to come from Denisovans into the ancestors of Tibetans. So it's quite fascinating to think that we would actually probably not have so big populations in Tibet today if it wasn't for this contribution from Denisovans. There is now almost every month coming out papers sort of pointing out other things that come from Neanderthals and Denisovans and our medical relevance. Just one example from another group in our institute. There is a group of 3 genes here on chromosome 4 that both in Asians and Europeans have variants that come from Neanderthals and Denisovans. They are quite frequent. Up to 50-60% of individuals carry them. And they are actually associated with medical conditions. So if you look on Helicobacter pylori, a bacterium that infects you in your stomach and can give you ulcers, you find that these variants from Neanderthals and Denisovans gives you increased resistance to this infection. So if you don't have an ulcer, you may perhaps thank the Neanderthals for that. But these same variants actually increase the risk for allergic diseases. So if you have allergies, you may perhaps blame the Neanderthals for that. So it's beginning to be a picture where these modern humans come out of Africa, meet Neanderthals and Denisovans that have lived in these other environments for hundreds of thousands of years. They mix a little bit with them, pick up some genetic variants that may have been of an advantage in that environment, and those variants increase in frequency. So it is something that we call adaptive introgression when we study animals that one wasn't so aware of might go on with present-day humans too. So finally then though, I think we can also begin to ask what did not come over from Neanderthals? What did we not inherit from them? And that may be even more important. So what we can do now is look over the whole genome. These are different chromosomes of humans. In red, the contributions from Neanderthals in Europe, and in green in Asia. But we can then look for parts of our genome where we would statistically expect Neanderthals to have contributed, but we don't see any such contributions, although we would expect it. So you can then ask, what is in those regions, and why are they interesting? Well, we could imagine that sort of genetic functions unique to modern humans would hide in those parts of our genome. Why are these functions interesting? Well, I think that there are actually things that changed in modern humans since the split from Neanderthals. There are things in our history that are really unique to modern humans. If we think, for example, of Neanderthals that lived for 400,000 years, maybe 4 times longer than modern humans have been around, there are stone tools at the beginning of their history and in the end of their history look, to me, who's not an expert, quite similar. An expert can see the differences, but they will have to explain to me what are the differences in those stone tools. Modern humans have been around for 100,000 years, and you actually don't have to be an expert to say that our technology today is different from 100,000 years ago. So our technology changes very rapidly. There are also things like depicting art that comes only with modern humans. There is, again and again, sort of suggestions of Neanderthal art people find. But if you sort of excuse me, it's sort of very modern art you find. It's some parallel lines on a wall or something like that. But things that really depict something comes only with modern humans. And of course modern humans are the first to spread over the whole planet and not be hundreds of thousands of individuals at any one time, but millions and billions of individuals. So we can now make a catalogue across the genome of everything where present-day humans differ from Neanderthals. So you can look for things that we all share today, no matter where we live on the planet, but the Neanderthals look like the apes. So if we're very strict and say, as far as we know, 100% of people today should have these changes, and Neanderthals and Denisovans should look like the apes. That catalogue of genetic changes is not very long. It is actually around 30,000 changes in the genome. So you can look through it in an afternoon in the computer. The only problem is you have very little understanding of what you see. You are extremely bad in predicting the functional consequences of any particular change in the genome. But just to give you a feeling, there are only 96 changes in proteins, for example, in this catalog. Here are all the proteins. One can start thinking about what could these changes mean? And I think that's the challenge for the next 10 years in this field. We're of course particularly interested in genes and proteins that may have a function in the brain, because we think something cognitively is unique to modern humans. And just then to end up on saying, how would we study these types of changes in the future? Because they are unique to modern humans. So I've gone around for some years to joke and say what we want to do is to make Neanderthal genetic variants into transgenic humans today. And we want to put human alleles into transgenic chimps and study the functions. And we can't do that for many, many reasons. Above all, ethical reasons, of course. There are now even more crazy people that go around and say we should take our list of 30,000 genes and actually make them all into a human baby and clone a Neanderthal and look at how that person would function. And that's, of course, for many, many reasons. That will also never happen. But it's not just a joke in a way. How will we ever really show that something that affects a human-specific function is really there? I think there are some things we can hope to do in the future. One is to say that in a future where in the developed world everyone will have their genome sequenced in the hospital, we'll have millions and millions of individual genomes sequenced. And the genome is of course small enough, 3 billion places, and every baby that is born, as I said, has something like 100, 150 new mutations. So every mutation compatible with human life is out there in the population, but they're generally very rare. But in the future, one will then begin to find back mutations to the ancestral old state. And I think it will become possible to study such individuals, probably. But that's a bit in the future. You can, of course, now engineer in these changes into human stem cells and study cells in tissue culture, and you can put these changes into the mouse. And I just can't stop myself for the last 2-3 minutes to show you our latest experiment in that direction. It concerns a gene on chromosome 7 that we studied for many years. It's called FOXP2. It's a gene that became famous because in a family in England over 3 generations, there's a severe language problem, a specific problem with language that segregates, and it is due to a mutation in this gene. This gene turns out to have 2 human-specific changes here, where humans differ from apes. So we were, of course, very interested when we started getting the Neanderthal and Denisovan genomes about these 2 positions. How did they look like in humans or like in apes? And when we started getting then the Neanderthal sequences and the Denisovan sequences, they turned out to look like humans today, not like the apes. So this then means that these changes happen back here. These changes are still interesting, of course, but they are shared with Neanderthals. So what you can then do is to take a mouse and engineer these changes into the mouse FOXP2 gene. And apart from one other change here, you then have a mouse that from its own FOXP2 gene makes the human protein. And you then have a mouse that you want to study its linguistic abilities. You try to speak to the mouse, and it will not answer. Probably it has a lifetime that's too short to really learn language. So, but you have an animal model anyway, and you can begin to then study it. So for example, we found that these humanized mice have longer dendrites, so longer connections to neighboring neurons than the wild-type littermates born by the same mother. And that's particularly in regions of the brain, circuits in the brain that has to do with motor learning motor behaviors. So one graduate student in the lab, when she finished, went on to study learning of motor behaviors in these humanized mice together with a group at the MIT in Boston. So they did experiments where the mouse is in a maze and have to learn that it should go towards a signal here to get a reward, chocolate milk, which the mice like very much. So the humanized mice and the wild-type mice learned equally fast to go towards the signal. But it comes a time then, if you always give the signal, say, to the left, where the mouse have internalized, I always go to the left to get my chocolate milk. It doesn't matter, you can take away the signal and the mouse will continue to go to the left. It turned out that that switch goes much, much faster in the humanized mice, learn this in 7 to 8 days of training, whereas their wild-type littermates take 11, 12 days. So they are a lot quicker in sort of automating muscle things that they have learned. And I'm not a neurobiologist, but the neurobiologists sort of tell me, think about, for example, when you learn to bike as a kid. In the beginning, you think about what you do. If you lean to this side, you sort of steer to that side and so on, and you're very bad at biking. Then comes a point where you suddenly stop thinking about what you do. You've sort of made it all automatic, and at that point, you get very good at biking. And that is this switch to sort of proceduralization, automation of something motoric you have learned. So the current hypothesis is then that these two mutations change something in these circuits in the brains that make you better at automation of muscle movements. And it's of course very tempting to then speculate, to say that the most sophisticated muscle coordination we do in life is articulation. If you think about it, I need millisecond control of what my vocal cords do, my tongue do, my lips do to produce articulate speech. So it's possible then that this has something to do with some aspect of speech and language. But of course many, many other genes involved in speech and language, but this might be one aspect of it. So it's encouraging, I think, that you can perhaps understand some aspects of these human-specific things in a mouse model. And I think you can imagine that you study more of these genes in that way in the future. So then to end, I hope that I then convinced you that if you're interested in human evolution, particularly recent human evolution, it is quite useful to have the genome of our closest relative, Neanderthals, because you can sort of distinguish what happened earlier on the human lineage and more recently. But I think that the big challenge for the next 10 years now is really to not just look at these changes, but study them functionally. And the way to do that will be in tissue culture, in cells, but some of it will also be done, I think, in humanized mice. And should then just end by saying many people are involved in this, many more than I can mention. I will mention one person in the lab, Matthias Meyer, who developed an ultra-sensitive way of doing DNA libraries. Without his work, we would not have these high-quality Neanderthal and Denisovan genomes that we now have since 2 years. And many people have helped analyze the data, particularly Janet Kelso, who coordinates all the bioinformatics in our group. And for population genetics, Monty Slatkin at UC Berkeley and David Reich at the Broad Institute. And with that, I thank you for your attention.